very familiar with James's pioneering
I'll click it.
pioneering research on the immunomarking procedure, which has gone through 2 or 3 different generations of research, which he'll be talking about today. I first met James. It took me a little while to meet him. We'd already been doing some protein marking work in my lab. But then I finally got to meet him at a symposia out in Arizona. And I think we'd already had multiple conversations on the phone at that point. So about protein marking and its different applications and the different ways to use it. So now, James, I did get your seminar if you want me to run it through here.
Go ahead. She got
Is that easier?
Go ahead. I'm having fits with it here. Can you do it?
I will go ahead. Yes, I will absolutely go ahead. Are you in a good space to get started?
I didn't hear a word you said. Your voice was clipping out. Both you and Cheng Lu's voice was clipping out. But I think we're good now. If you can operate the screen.
I will. Just tell me when to advance each time. Okay.
Yeah, I'll do my best. Sorry about that, folks.
Technology is great until it doesn't work.
True. There we go.
Okay, so let me just put this into full screen mode. And is that the right presentation format, everybody?
How about delete the?
Yeah, I'm trying to figure that out. Hold on. Swap displays.
Use F5.
And you need to be in presenter mode.
Yes.
Is that right? Does that work?
That looks good.
Okay. Well, I can see what's to come.
Are we ready?
We're ready, James, if you want to get started.
I apologize. I was having trouble. I was meeting with Cesar earlier today too, and I was kind of fumbling through this. I apologize. I'm not sure what's going on. I have done this before, but I've never had this kind of trouble. Again, I'm James Hagler. I'm coming from my home in Chandler, Arizona, but what you see right now is an overview of our beautiful research facility located in Maricopa, Arizona, which is about 40 miles due south of Phoenix Sky Harbor Airport. And we're out in the middle of nowhere in the township of Maricopa, surrounded mostly by Native American land. We're huge in cotton research. And there you see our beautiful facility that was built in 2006. Go ahead, Ann, next slide, please. So the goal of the presentation today, and let me first start saying that my favorite talks to give are departmental seminars. When we go to entomology meetings, we typically go and give symposium talk to the same old people, same old faces, and preach to the choir. So it's kind of fun to come and give general presentations of my research, and hopefully it'll spark some ideas from those of you who are not familiar with the protein immunomarking technique. So I'm going to give a general overview of the technique, the protein immunomarking technique, or I've coined the term the PIT. And I want to provide examples of both how this technique can be used for dispersal research and gut content analysis research. The latter of the two there is sort of a new concept that I think is really going to gain ground in the years to come. Next slide, please. So just a general overview. Next slide. Protein immunomarking, or any insect dispersal study, as many of you well known, there's basically 3 components. You got to apply the mark first. And there are several different ways I'm going to be discussing on how that is done. There's some subtleties involved. And then you ultimately got to capture the insects out in their habitat and then bring them back to the laboratory, and you detect them for the presence of the mark. Next slide. And so there are 3 basic types of dispersal research. There's mark release recapture research, or sometimes often referred to as central point release research. And there's instances where insects might self-mark themselves. And of course, there's mark capture, where this is more of the landscape level ecology research. And these three different types of research, the way you apply, capture, and detect the protein differ based on whether you got a mark release recapture, self-mark, or mark capture study. Next slide, please. So application of the mark release, for mark release recapture or central point release, the idea is you collect insects in mass or you rear them in the laboratory and you bring them back and you can mark them. Next slide, please. In some sort of vessel. And so, you know, if you have a nice hardy insect like a ladybug, there's all kinds of things. It's a real simple process. You could literally just pipette the protein mark on them. You can use a good old-fashioned Windex spray bottle, or in some cases we've used even like airbrush paint sprayers. So, you know, that's obviously pretty evident. But what happens if you have to mark a parasitoid? None of these techniques would work for a parasitoid. And what you see here in the circle is a whitefly parasitoid. And obviously, when we first tried marking them with the Windex spray bottle, it was a complete disaster. We drowned all of our specimens. So we had to get a little bit clever here and try to figure out a way, how on earth can we put a mark on that insect? Next slide. One way we figured out, and it was pretty simple, is we could spike their diet with the protein mark. And here you see a parasitoid of some sort lapping up some sugar water or honey drop, I can't remember which, that has been spiked with the rabbit IgG protein. So in effect, as they're feeding on this food source, they're taking the markup into their gut contents. So what you have here, basically using this technique, is an internal marking procedure. And that works quite well for the parasitoids. Next slide. But I also devised a way to mark them externally with a medical nebulizer. And when I came up with this idea at the time, my oldest son, he was very young and he had asthma issues, and we had to get inhalers for him. And that's where I came up with this idea, where you can put a little bit of the marker in a medical nebulizer and just pump air through the nebulizer. And what you see there in the right-hand corner is a Tupperware container that contains about 50,000 parasitoids.
No.
And
No, I'm in my office.
I'm hearing somebody in the background. Can everyone mute, please? Thank you. And we're marking parasitoids using this almost like a
No, I don't.
fog light,
I don't have
a fog light
I
device.
don't have anything for you to do right now. What do you got going on? Okay. Yeah. Yeah, I would just do that if you can. How are you progressing? Yeah, I know. I know. It's not. It doesn't look like it's over yet, is it?
Yeah, please mute.
Hey, Matt
Oh.
Bickerton, you're unmuted. Could you please mute? All right, James, did I go forward one slide? I'm sorry. All right, James, I think we're good. Do you want to keep going? James,
Can you hear me now?
we can
Yes.
hear you. Go
Good.
for it.
Okay. Okay. So that was some of the techniques used for mark release recapture, where, you know, you could just air blast them or blast them with a spray bottle, or you might have to get a little clever with some subtle techniques. Now for self-marking, self-marking is not that common of a dispersal type research. It's typically done with social insects, and we've had enormous success self-marking insects. And what you see here up in the upper left is honeybee colony, and the bees are pouring out. And as they're pouring out, they're getting dusted with day glow dust, green, as you can readily see. But what you can't see in there is I also have a protein powder, a milk powder in there. So they're actually being double marked with day glow dust and the protein. And the question might be, well, why would you want to mark them with both? Well, this particular experiment, we were trying to mark 8 different apiaries, each with a unique mark over a 5-mile area. And there's only so many colors of day glow dust that you can buy. So we created sort of a factorial marking system where some of the apiaries were marked with day glow green only, or they had day glow green and egg white, or day glow green and milk powder. Therefore, we had 3 unique marks. Just use those markers. And when we collected the bees from the field, we could come back, and if they had day glow green on them, then we could then assay them for the presence of one of the 2 marks. And that worked extremely well for marking honeybees. And then, of course, you could mark ants, as you see in the right-hand side of this some sort of bait, and you see a sugar bait of some sort with the protein mark in the bait, and they're basically self-marking as they feed on that source. And I worked with Paul Baker at the University of Arizona marking termites, subterranean termites. And what you see to the right there is a corrugated cardboard that we just literally soaked in rabbit IgG protein solution, and then we put it into the ground, and the termites would find it, feed on it, and then we could collect the termites at various distances from kind of their main source colony. And that, again, to my amazement, the technique worked really well for marking termites. Next slide, please. And I've been working with the Utah Bee Lab at the USDA on marking various bees, and here you see a bumblebee foraging. And the upper slide, there's some day glow pink dust with egg white mixed in it. You can actually mark the potential visitation site, and if there's a bee comes and visits, I think that's a lavender flower there, it will pick up the mark on its body, and when it returns to the hive, you could collect it and assay it for the presence of the mark. And the work we did with Natalie Boyle and her colleagues is we didn't want to kill these precious bumblebees. So when they returned to the colony, we figured out a way to collect them and then kind of give them a quick shower and try to wash the protein mark off them. And then we released the bees. So it was a non-destructive sampling technique, and that worked quite well. So you can actually mark the pollen or nectar sources where you think potential pollinators might be visiting. Next slide, please. And then in another work I did with Natalie Boyle and her team was the blue orchard bee, where we marked their pupil cases. They were rearing them in the laboratory, and you could just roll them in some protein dust. And as they chewed out of their protein case, they would pick up the mark. And again, that worked well. So these are just some ideas of the versatility of the protein marking technique for perhaps marking insects that are social in nature. Okay, next slide. Now, where the protein marking technique really took off is when we started developing methods to for mark capture research, because frankly, before protein immunomarking, there was really no good way to apply a mark across a landscape. And so over the years, and Anne's had a lot of success in this area too, of using conventional equipment, spray equipment, to mark insects, directly in their habitat. Next slide. And here you see my technician of 30 years, Scott Mackley. He's going through a strawberry field marking alfalfa trap crop embedded in the strawberry field. And I'm going to be talking a little bit about that research more in a few minutes. But he's just using a 3-gallon backpack sprayer. He could mark probably 100 meters of that alfalfa strip in 1 minute. Next slide. Here's my PhD students from Kansas State and Oklahoma State marking a canola field, and they got a little clever. They, excuse me, they created kind of a man-made boom device there and just simply walked up and down the canola spraying for natural enemies in an open field there. Next slide. And this is work I did with Rodrigo Krugner and Marshall Johnson, Russell Groves in citrus 10 or 15 years ago, just using a citrus sprayer. This was done in Riverside Ag Ops Farm, marking glassy wing sharpshooters and just air blasting the whole farm with this egg white marker. Next slide. And there's our conventional cotton marking device. Been working with Peter Ellsworth on that. Next slide. And this is Jimmy Click, my PhD student at the time at Oregon State, and he's air blasting for spotted wing Drosophila in some sort of berry field. And he had enormous success looking at movement of spotted wing Drosophila from natural habitat into their orchards. Next slide. And then Francis Sivikoff and Jay Rosenheim did the extreme marking. Here they are using aerial applicators to mark entire alfalfa field with the idea of we mark the whole field, and there's a huge field, and then the next day the grower came in and harvested the alfalfa and in effect created a dispersal event. And then we sampled like crazy in the periphery of the alfalfa field, mainly into cotton agroecosystem, to look at lygus bug and their natural enemy movement into the nearby cotton fields there in the California Bakersfield area. So that's the whole wide range of different techniques, the versatility of the marking technique, from marking minute parasitoids to marking entire landscapes. Next slide. Okay, capturing the insects. Now, again, if you're mark, release, recapture, or mark capture, or self-marking, again, there are different methods to capture the insects. Okay, and next slide. And a good old-fashioned sweep net. Here we are sweeping for honeybees. Telling you about that honeybee project a little earlier with Larry Tuber and Shannon Mueller in the Fresno area, looking at the long-range dispersal of honeybees in a conventional alfalfa versus a GMO alfalfa agroecosystem. Good old sweep net is pretty much my favorite method of collecting. Next slide. And here's Francis Scheller and Scott Mackley using vacuums. Francis is using a modified air blower. Just inverted the manifold on it, and she's collecting the natural enemies in cotton. And Scott there, if you look very closely for you students of entomology, he has got an original D-vac on his back, the Dietrich vac. And that's probably an entomology heirloom in my laboratory. And it's a monster, but it's highly effective. And some of the older field ecologists will really appreciate that piece of instrumentation he's got on his back. It only weighs about 50 pounds. Next slide. And for my parasitoids, I came up with a clever way of marking parasitoids. At least I think it was clever. We didn't want a big old vacuum and then have to go looking for a parasitoid in the sample. So I created a passive suction fan trap. I just went down to the Hobby Lobby and bought one of those model those little portable car engines, and pulled the wheels off of it and stuck a fan blade on the end of it and attached it to some D-cell batteries and created a suction with the fan blade. The original fan blade I made was a smashed can of Sprite soda, and I cut with shears and glued it with shoe goo and then embedded it all inside that PVC. And then that turns out that dram snap vial on the right fits perfectly in that PVC. And you can cut the bottom of the vial out and put organdy fabric, and you can slide that in and out and cap it ultimately and collect parasitoids. And it was amazing how well that worked. And you could lay those things, those fan traps out in a grid and sample like crazy for those whitefly parasitoids. And it was very effective. Next slide. And then the bane of my existence is the sticky card. I encourage you all to avoid sticky cards. It's a nightmare working with these things. But having said that, there are instances where it is necessary. To the left there, we're sampling, it looks like in a alfalfa field there, and you can see we've collected thousands of insects. And then on the right there, we've got some traps. If you look in the background there, they're on poles. I worked with Jackie Blackmer, the late great Jackie Blackmer, on the project studying glassy wing sharpshooter movement, and we were able to use the marking technique and look at glassy wings and see where the strata in which they tend to fly from one area to another. And sticky cards were really the only method we came up with for that particular project. And to my surprise, the stickum doesn't seem to affect the integrity of the ELISA procedure. We were still getting good marker detection, even with these terribly sticky insects. Next slide. And then ultimately, you bring all your insects back that you collect in mass and you start the assay process. And it's basically a 4-step process. I have developed several ELISAs over the years. It's a 4-step process. And if you could just hit next slide a couple times in a row, you add your antibody, you add your insect sample, and then your secondary antibody. In this case, this is the sandwich ELISA format. And then one more click there down, and you add a substrate, and then you'll get a coloration reaction. Okay, one more N, and we'll have this one done. And you get a nice positive reaction in a, this is not an atypical response in an ELISA. We see the nice blue response there. It's a positive for that protein mark. Okay, next slide. And just a few ELISA fun facts, if you will. Over the years, I've developed 3 ELISAs. 3 are for mark release recapture. I call these the first generation markers. And then the second generation was, they were developed for mark capture. And they're, the mark release recapture ELISAs are sandwich ELISA format, which have some pros and cons. And then the mark capture technique is in the indirect ELISA format, and it has some pros and cons, again, depending on the type of research you're doing. The main thing about ELISA, which I really like, is it's a simple procedure, and with a little training, you can train someone at the beginning level, even high school, a high school intern, college undergrad can learn the technique in a couple days in the lab. You can process the samples by the hundreds, thousands per day. We're up to 2,000 a day if we have to, and that's just 2 people. And it's about 10 cents a sample, maybe a little higher now. I'm not sure when I came up with this calculation. And it's sort of a quantitative assay, even though really we're only interested in the qualitative aspects. Next slide, please. And what I mean by quantitative assay is you can actually get readings on these different blue intensity wells, and you can create a critical threshold value as shown in the dotted line there in that graph, in that fake data set I have. That dotted line is basically the mean and 3 standard deviations above an unmarked insect. And what that does is that you can create, throw out the human error component and look at the data with hard numbers and get a little bit better handle on what you call a marked insect and what is not a marked insect. Next slide, please. Okay, so that's kind of just a general overview of the protein immunomarking technique. What I'd like to show now is a couple examples, kind of a primary example of my dispersal research and of my most recent predator gut content analysis research. So let's go to the dispersal stuff first. Next slide. And I'm going to give you about a 5-minute presentation of my favorite study, field mark capture study to date. It's trap cropping on the California Central Coast. And if you look, what you're seeing here is the world's largest, or at least at the time, the world's largest organic strawberry field located in Watsonville, near Monterey, California. It's one of my favorite projects because we got to do it in the summer months, and it got me out of Phoenix, Arizona, and to Monterey, California. Got me out of the 115-degree cotton fields and into about a 70-degree strawberry field. Next slide. And I was brought into this project with my dear friends Sean Sweezy, Diego Nieto, and Charlie Pickett and Janet Breyer to mark strips of alfalfa that are embedded within the cotton field, the strawberry field. Next slide. And if you look at this field, you'll see every 50 rows of that strawberry, you'll see a strip going down there, and that's the alfalfa. And the idea is to get blooming alfalfa growing out there to lure the major pest of strawberries, and that's the lygus bug. Next slide. So this is quite a simple process. Here's Scott marking one of the central rows of alfalfa, and to both sides of it, you'll see a beautiful strawberries growth going on there. And if you look in the background there, 50 rows over, you'll see another strip of alfalfa. Next slide. And our sampling scheme is we took one of the central zones, which we call the mark zone, and we marked it, and then we sampled 1, 2, and 3 rows to the east and west of the strawberries, and the 10th row. And then we went over to the adjacent strip crop that was unmarked and sampled the alfalfa in those, we sampled for insects in those alfalfa rows. So it's a pretty simple sampling scheme. Next slide. And we sampled by using the vacuum. And earlier I told you, SweepNet's my favorite, but there's instances for every study you have to take in various aspects, modify it. And then we had to modify it to a vacuum here because you wouldn't want to be taking a SweepNet to that precious strawberry. That's very valuable commodity to the growers out there, and we didn't want to be beating up their strawberries. So the suction vacuum shown here was the best way to cause no damage to the growers' farm. You always have to take things like that into consideration. So we sampled like crazy after marking. Next slide. And so the question was, do alfalfa trap crops lure the lygus bug? So here's our sampling plan again, and you will see indeed that the lygus populations are much higher in the alfalfa, as you would expect. And then we took all of those specimens that we collected and we ran the ELISA on them for the presence of the egg white mark, which we put out. Next slide. And the black bars filling in the total show you the proportion of marked insects that we obtained from this study. And what it shows you here is that after 1 week of sampling, we sampled roughly, I think we sampled every other day for a week. So 1, 3, and maybe 6 or 7 days after marking, roughly 80% of our insects we were collecting in the mark zone were still marked. So that's a pretty good sign. And you can see very little spillover into the adjacent strawberry rows. So it kind of gives you an idea of the retention of this alfalfa strip crop. Now, one thing about the marking technique, and I encourage all of my students who, we typically have an insect that we're interested in, but I tell them by all means, in particular, this was a ligas study, collect all the insects and save them all because, next slide. Well, let me back up a little bit. Just say, in summary of that, roughly 6% of that acreage is dedicated to the trap crop, and we were collecting 90% of the ligas in that 6% of acreage. Okay, now next slide. So I always encourage when you do a study, say for a ligas pest or whatever pest of interest you have, it doesn't hurt to go ahead and save all of your specimens because you're marking the ligas bug true, but you're also marking everything else out there. And it gives you an opportunity to branch out and look at various aspects of the predators or even the parasitoids. Next slide. And so the question is, what does this trap crop do for the natural enemy complex? And so for like the minute pirate bug, which is by far the most common natural enemy in a strawberry field, you can see not too much, there's not any really pattern there. It seems like the ligas, the aureus are distributed evenly across the field, which is fine. Next slide. And you can see that again, over the duration of the study, they were pretty well retained in the middle row there where we marked. There was some kind of a bell-shaped curve out 3 rows to either side. Excuse me. Next slide. And of course, then with ladybugs, we found 3 different species of ladybugs, and they were very dedicated to the alfalfa. Next slide. And they retained well, and it looked like, in fact, there was some movement of the ladybugs to the east, which would make some sense. In this field we were in, there was always a pretty good breeze blowing off the Pacific Ocean coast there, and our guess was, you know, maybe they were, when they did take to flight, they were taking the downwind path. Next slide. So the beauty of this experiment, or the research here is, you know, here's our kind of our IPM pyramid, if you will. Next slide. We had several of the components. We had cultural control going on for us here in the form of strip cropping. Next slide. What I didn't have time to show today was they actually used some mechanical control, which is this super duper insect vacuum. Again, 90% of the ligas are in that strip crop, and there are instances where the grower will take a vacuum and suck up all the ligas and physically remove them from their field. They don't do that as much now as they used to, but it is certainly, they definitely have that capability. Next slide. And then we had other studies, you know, kind of looking at both the predator and parasitoid complex, looking at both their dispersal capabilities, but I also published a paper in 2019 looking at the gut contents of many of these predators using the PCR gut assay technique, and did show, in fact, that as you would expect, predation rates or frequencies were much higher in the patchy alfalfa than they were in the strawberry rows. Next slide. And the best part about this whole experiment is, you know, it is an organic farm after all. No chemicals were needed based on this cultural control tactic. Next slide, please. So in 2019, Jana Lee invited me to write a paper on this technique and called it Supermarket. And it's virtually got all the papers published to that date on the technique and all the recipes and all the caveats of the marking technique that I had briefly gone over today. So now I want to switch gears here. I think I'm doing okay on time. I realize we started a little bit late. I want to talk about a universal food marking technique for gut content analysis. And I actually discovered this technique. My undergraduate student, Christy Duran, needed a senior project, and we kind of just did this literally in a petri dish and managed to get it published in 1994 using the marking technique for gut content analysis. Because if you think about it, if you mark an insect and another insect comes along and feeds on it, bottom line is you could probably detect that mark in the gut of the predator. Next slide. So I want to talk a little bit about a gut analysis technique for pinpointing egg-specific predation events. And this is done with Ayman Mostafa, who was doing a sabbatical in my lab. And our target prey was Ligus hesperus. Next slide. But more specifically to the Ligus hesperus egg stage. And here you see some Ligus eggs embedded in parafilm. Next slide. And the Ligus egg stage is a difficult prey item to examine because Ligus exhibit what's known as an endophytic oviposition behavior. And what you see is basically an endophytic behavior is the Ligus oviposit their eggs into the main stem, typically the main stem, say of a cotton or alfalfa plant. And then they cap it with this almost like a manhole cover cap. That's a tough cap. So most of the egg is actually embedded in plant tissue. Next slide. And people, of course, have hypothesized that it could be a survival mechanism for the Ligus egg to keep it away from like abiotic factors such as rain or biotic factors such as predator, or more than likely a combination of both. But you can see where it could serve as a protection evolutionary process, protecting the egg from predators and abiotic factors. Next slide. So the hard part of this whole study was simulating a Ligus oviposition event, trying to figure out how to get a mark on a Ligus egg. And we have a colony of Ligus being reared at our laboratory on artificial diet. And what you see here is a oviposition pack that's made out of agar that's been poured into a parafilm packet. And then Ligus simply go and oviposit into the parafilm. It simulates plant tissue. They won't oviposit anywhere else. They need that little bit of a tug, if you will, of a plant tissue or an agar or the parafilm. And we can modify that egg packet to where they only lay little patches of eggs. We put basically masking tape on the good portion of it, on that one on the left, and created little strips of Ligus eggs. Next slide. And then I created an artificial stem by taking a wooden dowel and putting a notches in it just to give it some space. And then we took those parafilm strips, and the parafilm almost acts as a Band-Aid in texture. And what I could do is then take that endophytic egg. Well, let's start with the exophytic egg, the part A there. And I just inverted the eggs so they were fully exposed in that artificial stem. And then the endophytic eggs we put where the egg was embedded within the notch of the stem. So in effect, we created 2 egg exposure treatments to study whether predators, whether this oviposition behavior is protective of the predator. So we had an exophytic sham control, and those eggs I marked with rabbit serum, and then we had an endophytic natural treatment, which I marked with chicken serum. Next slide. And we put them in field cages and exposed them to either lady beetles or colops. And each cage contained, I think it was 20 individuals, okay? and we did 20 cages. Next slide. Of each species. So each dot on this is a experimental unit of a cage. So like if you see that ladybug, where the letter C is, it's around 60%. That means 60% of the predators in that cage, which there was an N of 20, were positive for the presence of that mark on the exposed egg treatments, just to clarify. So in theory, there should be 20 dots under each of those bars, and that's a experimental unit of 20 insects. And what you'll, a quick glimpse, you'll see for the ladybugs, they weren't as efficient predators as the Colops beetle. The Colops beetle was a pretty darn good Lygus egg predator. And what you'll also see is that the exposed treatment was more vulnerable to both species, which shouldn't be too surprising. Next slide. So this study showed that the endophytic eggs are less vulnerable to predator attack, and that Colops is probably in a natural setting going to be a better egg predator than the lady beetle. Next slide. Okay, let's take it to the next stage, and I'll wrap up showing a paper that just came out in press. I think it might be December issue, I should say, of the Journal of Insect Science. We took it to the next step where we started looking at stage-specific predation events. And so for the eggs, I marked them with rabbit IgG. Again, our target was going to be Lygus. And I took Lygus nymphs and marked them with chicken IgG, and the adults are marked with rat IgG. Next slide. And this study was not done in the greenhouse cages, it was done in the field. Next slide. And we went out the day before the study and draped a organdy cage at the base of a cotton plant. Now, this is a Vernonia plant, but this is the best spot I had of the draped cage. And this allowed the natural enemies to regather. And then we came out the next day. Next slide. And we carefully went in there and put up an artificial stem with our marked Lygus eggs in their endophytic state. And then we came back a few hours later. Next slide. We pulled up the cages, and then we dropped in three marked adults and 3 marked nymphs. So we had a fairly realistic population of marked Lygus in these cages. We had 3 marked adults, 3 marked nymphs, and then we had a small patch of Lygus eggs. And we put up 44 of these cages. Came back 6 hours later. Next slide. We cut the cages at their base. Next slide. Well, there they are laid out down the cotton field. And this cotton field, just beyond those trees, is our laboratory. So at the end of we did this first thing in the morning. At the end of the day, we came in and snipped each of the caged cotton plants at their base, threw them in the back of our truck, and hauled to the laboratory and stuck them in a -80 freezer to kill all the insects immediately. Next slide. And then we simply went through all the cages and counted. And what I like about this open field cage technique, it gives you a per capita estimate of the predators on a per plant level. And you can see, next slide, we collected 760 predators in the 44 cages. And next slide. Next 2 slides, actually. And they were dominated by the big eye bug, Geochris, and the crab spider, Mysumenops, were our 2 predominant predators in this particular system in our cotton. Next slide. Now we took all 700 plus of those specimens and we assayed it for the 3 we assayed it 3 times for the presence of the egg mark, the nymph mark, and the adult mark. Next slide. And here's the synopsis of our research. The Colops beetle, if you recall, was a voracious egg predator in that one simple study I did earlier. Well, sure enough, it turned out to be a pretty good egg predator in the natural cage, the field cage as well. There was one predation event on a nymph, but you could pretty much say that the Colops beetle was a stage-specific egg predator. Ladybugs didn't do very well. Neither did the Schimmus, which is another coccinellid, and the C7, which is a coccinellid. They really didn't score well in the ELISA. So, what of the beetles, it looks like Colops was the main biological control agent, and it was targeting the egg stage. Next slide. And for Geochris, or the true bugs, I should say, our true bug population in cotton just generally is these four characters here: the big eye bug, the assassin bug, aureus, the minute pirate bug, and then of course the nabids. And you can see the aureus and nabis were not effective on ligus. The Geochris, again, looked like it was an egg feeder, and it actually fed on a few of the nymphal stage as well. So basically here, what I'm showing here, I think perhaps for the first time, is a gut analysis technique that we can almost call quantitative results. Given it's a field cage, I'm not going to go that far. You can see the ligus population is 44 cages. We had 1, 2, 3, 4, 5, 6, 7, about 15 ligus egg predation events. I may be a little off on that calculation. Any dot above that dotted line, that horizontal line, is a positive response. And you can see 3 predation events on the nymph stage. So it kind of is getting not only at a quantitative nature in gut analysis, but it's also doing something the PCR technique can't do, the gut analysis technique. And it's, it's a stage, we're looking at stage specificity as well. So that's a pretty big deal for those of us in the gut assay community, to have a quantitative or semi-quantitative and stage-specific assay. These are attributes the PCR at this point just don't have. Next slide. And then our crab spiders, not much going on. The crab spider is the only one, though, that did show a predation event on an egg, nymph, and adult stage. And then we had an invasion of fire ants into some of the cages, and the fire ants were dedicated egg predators, which isn't too surprising. Next slide. So the advantages of this so-called universal food immunomarking technique over the PCR assay: it's semi-quantitative, life stage specific, which is something I've been striving for a long time. You can detect cannibalism. I will say, and I didn't show the data, that we also assayed the ligus bugs that we added to the cages, plus the native population that was already there. And we did see some cannibalism of the adult on the nymph stage. Not a lot, but we did see evidence of cannibalism. And work I did with my undergraduate student, Gabe Zelnick, some time ago, we showed that this technique can be used for scavenging. So you can look at scavenging behavior. We learned that Colops is a notorious scavenger as well. And of course, this technique is very cost efficient. The techniques have already been developed. They're all commercially available. So there's really no PCR primer design involved. And the assays, as I mentioned earlier, the ELISAs are really well suited for mass throughput. Next slide. So again, Jana Lee asked me to write a review article on this technique, and you can get that in the Annals of the Entomological Society of America, where I talk about all the research using this technique. Again, I discovered it, developed it in 1994, but it kind of just sat on the shelf for almost 20 years, and I got it out again back in 2013. And I would say in the last 5 or 6 years, I've made more progress on gut analysis, I think, than I did in the previous 30 years using the monoclonal antibody and the PCR gut assay approach. I'm getting answers to questions that I could never get using that technology. And so I'm really excited about this technique for gut analysis. Next slide. And so in conclusion, I hope I gave you a general overview of the protein immunomarking technique. I hope I sparked some interest in how everyone in the audience might be able to use it for their, whether it's dispersal research or biological control research, looking at predator efficiency. I talked with Cesar's group a little bit earlier, again, about conservation biocontrol and some of the plant feeding characteristics you could look at too. It's not just for predation. If you're marking the plant, herbivore that feeds on that plant is going to pick up that mark. You can put it on the plant, you can put it in their nectar resources, you can put it on pollen, all these things. You can mark these things and do gut analysis research, not just of carnivores or the predators, but of herbivores and nectivores. Next slide. So that concludes my talk. Of course, this work wouldn't be possible without my A team. I've got a wonderful cast of characters working in my laboratory. This particular summer, last summer, Iman was shown there on the far right, was doing a sabbatical in my lab. And on the far left there is my technician of 30 years, Scott Mackley. This work couldn't be done without him and the wonderful cast of characters we have in our lab. The one thing I can say in our lab is, and I hope to pass on to you when we get into research, we have a ball, we have fun. It's been a fun ride. So I think I'll conclude with that. I know we got a late start. It's 10 o'clock sharp. I hope there's time for a few questions. Again, I apologize for my fumbling around with the Zoom. I'm not very proficient, obviously, at that, so I apologize. Any questions or?
Thank you very much, James. That was excellent.
Yeah.
Thank you.
Can ask question. Thank you.
So I'm going to stop sharing my screen here so you can see everybody. Okay. Do we have any questions?
I have a question, James. You mentioned the rabbit, chicken, and rat IgG for marking different like stages. What are the main differences between these different proteins in terms of, say, longevity or cost? Are they similar?
Okay. I'll tell you right now, they're all very expensive.
Okay.
They're very expensive. That's why we only, and for the dispersal work, we only use it for mark, release, recapture, where we can mark everything in the confines of the lab. For the predator research, which I think is your question, we use that mark because it doesn't take much mark to mark your prey items. So you can incur the expense, but the IgG assay developed to be detected by the sandwich ELISA format. And for gut analysis, the sandwich ELISA format is the only way to go because it is so much more sensitive. Now, your question is expense. A couple milligrams of that material can cost in the hundreds of dollars. Luckily, it can be diluted way out. As far as longevity, I'm not sure if you meant shelf life. If you buy it, if you keep it frozen, it'll stay forever. But I think what you meant was once they feed on the item, how long will it stay in their gut? And we've shown that it will stay in their gut for about 6 hours, regardless of which protein you use. And that's why the cage studies I conducted, it's so important to have that data prior to the study. So I knew when I threw my prey in that cage, I would have to get out there by the 6th hour to catch everything that might have had a predation event, or they might have digested it by then. Does that answer your question?
Yes, thank you.
The external marking, though, Cheng-Lu, can last for, I think it's definitely at 7 days for the food-based proteins. I haven't used the IgG ones, but we've detected it even up to 10 days under field conditions with rain events. And I apologize for my kids misbehaving in the background.
No, you're absolutely right. You know, when we're looking at the internal mark, we have digestion going on. So that's breaking it down. But I know you've had around a week to 10 days. And I've had studies like on ladybugs that have gone a month. And believe it or not, that termite example I showed, now granted that was the IgG protein, we sampled 30 days after we put that marker out there, and we were getting marks. So it has an enormous field retention. One of my initial fears 25 years ago is you put that mark out, and because it was a protein, that the UV light would break it down or whatever, and it just isn't the case at all. That stuff's incredibly stable in the environment.
James, one thing we've seen is that we do get a degradation of the protein marking over time. We're doing indirect ELISA, not sandwich, because it's all external marking, but I'll talk with you about that later. Is that it does seem like the insects will clean themselves. And so we lose some of the marking that way. It's not, because even in the lab, we get that and we're not having UV degradation. So, but after time, the insects will just kind of, in their natural cleaning behaviors, they will clean it off. So it's something
we will see that all the time. Yeah, we see the degradation over time, no doubt about it. And that's again why it's so important. I showed that mock data set with the ELISA critical value threshold there. And as time goes over time, that blue spots can get a little bit lighter, but when you have that optical density readings and you can create a critical threshold value, it gives you more confidence in what is a positive mark over time as well.
I have a quick question. The proteins that you use, I guess like they're binding and you talk about them being degraded over time. Is there something, at least internally, you could think about that would increase the binding affinity so you could detect them for longer periods of time? Specifically, I know you spoke about ants and eusocial colonies. Just based on any experiment I've done, they will groom themselves, and anything you have that disrupts the chemical cues will do that. So I was wondering internally if you could have something, maybe a different protein with a higher binding efficiency, would you be able to detect it for longer?
I suspect the answer to that question is yes. It'd be a matter of finding that protein and developing ELISA to it, though. That's a good question, though. And we've actually looked at even like for external marking, you know, in the field situation, trying to add something, you know, like when you spray insecticide, sometimes you add a surfactant to increase the kill power or the absorption of an insecticide. There might be some sort of surfactant you might be able to add, like Silhouette comes to mind, or I can't think of the other ones that are off, you know. But as far as an internal, if you're looking at ants, I don't know what that would be, but there could be something there to slow the digestion process down.
Yeah. I know a lot with ant colonies, they have kind of sub-functionalization of different proteins. So the proteins actually bind to different places on the body. So I wonder if you could like modify the proteins so it would bind somewhere and you could detect it for longer. And then it would kind of decrease the cost too, because it's, I mean, antibodies are super expensive, so.
Right. Right. That raises a good question. I read in a human physiology textbook years ago, because the question I had about these IgG proteins is how they last as long as they do. Now I realize I can't compare insects with humans, but they were talking about these IgG proteins in humans tend to bind to the gut. Kind of like what you're getting at, I think, with yours,
Yeah.
your question. So absolutely, there could be something that could bind and retain these particular marks longer. That'd be a fascinating area for research, future
Right,
research.
yeah. This
As I
was,
was telling Cesar's group this morning, go ahead.
oh, yeah, I was just gonna say, this is all like totally new to me. So I think it's like incredible that this kind of marking technique is being used. So I think I'm just like kind of thinking out loud, but it's very cool. I really liked your talk. Sorry, I didn't mean to interrupt.
Oh, thank you. Well, that's what I talked to Cesar's lab crew this morning, and it was a very exciting conversation because I could see light bulbs going off above their head. And that's why I kind of alluded to the fact my favorite presentations are departmental seminars, because if you look at my resume, you know, I'm a USDA employee and my mandate is cotton entomology. But if you look at my resume, I've been all over the board. And it's because people like you, when I give departmental seminars, will approach me. And I've worked with ants, honeybees, termites, mosquitoes recently, glassy wing sharpshooters in California, spotted wing in Oregon, and bed bugs at Ohio State. I mean,
Yeah,
and it's all as a result of departmental seminars.
Yeah, it's hugely applicable, but I always like the talks that get you to ask other questions about like, could you do this and in different fields. So thank you so much.
Thank you.
I think I will have to end now. Thank you, James, for excellent seminar. So
Thank you,
Thank
James.
you.
yeah, for students who registered for the class, we'll continue to stay here for another maybe 15 minutes. So everybody else can leave now. So we have a 5 minutes break.